You want to go ahead and begin? Yeah, there you go. All
Yes,
right. Hello, everyone. It's a pleasure to be here. Thank you so much for the invitation. It's my first opportunity to give a talk with an African audience, and I'd love to travel to Africa one day. So it's great to meet you all, and looking forward to chat with you. So today's
it'd
talk
be an
is
honor.
about our work on studying the process of translation of messenger RNA in mammalian cells. Just a few disclosures. I am a Sanofi employee and hold some financial stakes in the company. And this work was jointly funded between Sanofi and the University of Texas at Austin. So I want to introduce myself a little bit in the context of previous research in the simple diagram, which indicates the cycle of RNA, the life cycle of RNA. So RNA is born through the process of transcription, whereby DNA is transcribed into RNA. And that RNA is further processed in the nucleus and eventually exported to the cytoplasm. And here it can localize and be translated into a protein. The final fate of RNA is to undergo the process of degradation. So one pathway for degradation is called the microRNA pathway, and these are tiny RNA molecules that bind messenger RNAs and lead to their degradation. So a lot of my PhD work focused on this last piece of the pathway, studying how microRNAs engage their targets and initiate the degradation process. During my postdoctoral work at the University of Washington in Seattle, I started studying the process of transcription and how DNA encodes information to specify the amount of RNA that should be produced and in individual cell types or tissues. During that time period, I also studied mRNA processing in a step called alternative polyadenylation. And that term means something that defines the 3' end or 3' boundary of the mRNA. And so in mouse embryonic development, we performed single cell sequencing and characterized many of the alternative polyadenylation isoforms across early development. And then later in my biotech world, like I worked at Calico for a while. It's like a Bay Area in San Francisco. It's a small startup of about 250 people. And there I studied both the process of transcription, but also the process of mRNA degradation, returning to my roots in my PhD a little bit, trying to understand which molecular mechanisms lead to the decay or degradation of mRNA. But today I'll be talking about a new project that we just finished. It was like a 2-year project. It's my current job in Sanofi, studying the process of mRNA translation and how it's regulated. So at least some folks in this group might be more on the computer science side of things. So we've taken a lot of the innovations from computer science, specifically in deep learning and artificial intelligence, but applied that to study DNA in this context. So in this case, you can take a DNA sequence and a measured readout, like this cage readout, for example, measures transcription at every nucleotide in the entire genome. And with the deep learning, you can train a model to predict this cage signal from the DNA sequences. And early in 2015, the field started off just predicting a binary signal. That is, yes or no, is it a transcribed region? But as the field evolved over time in 2018 or so, there were some studies that were trying to not just predict yes or no, does it transcribe, but instead the continuum, entire continuum transcription from low, medium to high. And so far, this was at promoters or specific regions within the human genome. And again, around 2018, this work was expanded then to have higher resolution. So not just looking at gene promoters or a specific subset of the genome, but instead the entire human genome. So they just took bins across the genome, and across each positional bin across the genome and each chromosome, we tried to predict now the level of transcription in every single bin genome-wide. And this work matured further, and in 2021, people started predicting not just individual bins, but at every single nucleotide of the genome, the level of transcription. So over the years, the resolution of these models has really improved, and that goes hand in hand a little bit with computer engineering advances like better GPUs, better ability to fit large sequences in memory, and all this data and information. Now, when we train a neural network like this, one of the benefits or advantages is also being able to interpret what the neural network tells us. So in this example at the bottom, we've zoomed in one promoter sequence, and we can just ask the neural network which regions of this promoter sequence are activating transcription versus inhibiting transcription. So it would go a positive direction or negative direction depending on which region we're looking at. And so I participated in 3 of these studies. So Espresso early on in 2018, followed by Informer, and our latest iteration published earlier this year called Borzoi. But today I'll be talking about a different work that I've been carrying out at Sanofi with the team shown here. So this is my manager on the left, Fernando, and 2 of my team members, Dinghai and June, who led this work in collaboration with Logan, Yue, and Professor Jan Senik at UT Austin in Texas. So this work in particular relates to messenger RNA or mRNA. And mRNA provides the set of instructions to build a protein, but there are different regions of the mRNA. So there's what's called the 5' cap and 5' UTR, which are at the beginning of the mRNA that are regulating translation or the stability of the RNA. This is followed by the coding region, which is the actual region that creates the building blocks for the protein, but it also can have a dual roles in actually regulating translation and mRNA stability. And the final piece of the mRNA is this 3' terminal region called 3' untranslated region or 3' UTR, and the poly A tail. And this region is thought to regulate the RNA stability more so than other aspects. Now, the beauty of this type of therapeutic is that it can be used for many different types of therapy. So during COVID-19 pandemic a few years back, we saw the emergence of a really amazing new type of therapy called mRNA vaccines. And mRNA vaccines encode this an antigen so that the immune system can respond and recognize this and protect you from the virus if you get in encounter with the virus. But mRNA is very versatile. It can be used for many other things besides vaccines. You can use it for building enzymes, peptides, antibodies, or bispecific antibodies. These are sometimes used, for example, in cancer mRNA therapy. You can even use it for encoding nucleases, such as CRISPR. So some people use mRNA technology to engage in genetic engineering, for example, rewriting the DNA code to correct a mutation or disease-based mutation in the genome itself. So it's a really powerful type of technology that can be used for versatile applications. But much of our work at Sanofi relates to mRNA vaccines in particular because there are many infectious diseases around the world needing treatment. So our goal is to stimulate a nice immune response by expressing an antigen of interest. But if the antigen yield is too low, and that is the mRNA doesn't produce enough of the antigen, then it's very difficult to stimulate the immune response. So we want to have a high antigen yield. And antigen yield depends on 2 regulatory properties. One is the rate of degradation of the mRNA, or called mRNA half-life, and the other is the rate of translation of the mRNA, or translation rate. And each of these in turn depends on how that sequence is designed. So the coding region or the UTRs are regulating the amount of ribosomes that bind to the mRNA and how stable or unstable the mRNA is as well. So the goal of my team is to actually design the mRNA sequence so that we can better optimize these regions, improve the mRNA half-life and translation rate, and therefore have better yield so we can stimulate a more robust immune response. Now, several years back, before I joined Sanofi, I engaged in this project to try to predict mRNA half-life from the sequence of the mRNA. So what we can do is we can take the mRNA sequence from left to right, and each nucleotide here we can encode into a matrix. So A, C, G, or U. Just for every single position, we can encode which character it was. We also have the reading frame of the open reading frame for every codon, and the presence of splice sites in the sequence. And that's widely available and easy to download from online annotations. So that is the input to this model and everything that it's able to see. But then we feed it through multiple sets of convolutions to build a convolutional neural network, or CNN. And the first layer of a convolution usually learns something like motifs, for example. Like maybe, for example, a UGC motif might be relevant to regulating RNA half-life. So that's the sort of principle the first layer can learn. And as you get deeper and deeper into the network, it can learn greater abstractions, like combinations of motifs. If you put motif 1 and motif 2 together at a certain distance, then maybe that can regulate RNA half-life or stability. So that's the sort of principle this type of architecture, a neural network architecture, can learn. And when we trained this model, it actually performed very nicely. So on the y-axis is the observed half-life, and on the x-axis is the model prediction for sequences it had never seen before, so cross-validated model prediction. And we are able to achieve a Pearson correlation of 0.77, which is far superior than prior models that attempted to predict mRNA half-life. So this is the work that we did before, but another missing piece of this was to understand the translation rate now. So that was no one had really modeled exactly how to predict translation rate very effectively from the RNA sequence. So what exactly do I mean by translation efficiency or translation rate? So here's just an example of two mRNAs. You have one at the top that has only one ribosome loaded. It has maybe that ribosome moving slowly, and that leads to less efficient protein synthesis. In the case on the bottom, there are many ribosomes loaded on this mRNA, and that leads to more efficient protein synthesis. So the core question is, what is the difference between the first mRNA and the second mRNA that leads to low versus high rate of translation or protein synthesis? So there's a really cool technology that was developed maybe 10, 15 years ago now called RiboSeq that relies on this technology where you take an mRNA loaded with ribosomes, and those ribosomes could be actively translating in the cell. But if you freeze those ribosomes in that moment in time and digest with RNase enzyme, it will leave little footprints of the RNA that were attached to the ribosome. And in the late 1980s, it was discovered that just by doing that simple step, you can visualize the remaining band on a gel and see the short band that's about 32 to 36 nucleotides long. And those, it turned out, were what's called the ribosome protected fragments, or the little pieces of RNA attached to the ribosome in that moment in time. Now, if you take those exact fragments and visualize them on a gel, do a gel cutting between nucleotides 26 through 34 in length, and purify that subset of RNA, you can generate a high-throughput sequencing library with this RiboSeq technology. And if you look at the ratio between the number of those protected fragments relative to the total amount of RNA in the cell as measured by RNA-seq, that ratio will tell you about the rate of translation of that mRNA. So we call that translation activity or translation efficiency with this acronym TE that I'll be using throughout the talk. So we're not the first to actually evaluate this question. There was this very nice study in 2019 from Georg Selig's group at the University of Washington, in which his team tested over 200,000 random 5' UTRs. So they linked these to the same open reading frame, created mRNA from in vitro transcription, transfected that RNA into cells, and then measured the rate of translation with polysome profiling. Subsequent to this, now they had data for hundreds of thousands of different mRNAs for each of these 5' UTRs. They trained a neural network model to feed in the UTR sequence and predict the rate of translation from the measurements. And this model is very nice in predicting how effective 5' UTRs are, but it has some limitations. So one major limitation is that the only thing the model can see is the 5' UTR sequence. And it was actually discovered a few years later that if you take the same model called Optimus on the x-axis and look at its Pearson correlation in predicting different tasks, so if you look at RiboSeq, for example, shown in green here, the Pearson correlation in predicting the rate of translation from RiboSeq is actually not that good. So we get a Pearson correlation only about 0.2 to 0.3 or so at best. Which means that as great as this model is, it's not very good at predicting the rate of translation of endogenous mRNA as measured by RiboSeq. So this was a major limitation in this existing model, and so we tried to build something a little bit better than this current model. So our first goal was to ask, can we derive a more accurate measurement of translation efficiency by performing a meta-analysis of experiments? And here our goal was to systematically collect TEs for different endogenous mRNAs across many studies and cell types. Next, we asked, could we directly predict TE from mRNA sequence? And the goal here was to better derive machine learning and deep learning models to predict TE. Finally, we wanted to interpret these models and ask whether we could learn any mechanistic principles governing translation efficiency by evaluating how the spatial positioning and presence of different motifs affects TE. So my collaborator at UT Austin performed one of the most challenging steps of this entire work, which is to actually deeply curate thousands of different studies of RiboSeq data across the entire world. So his team collected over 200 studies in human and 100 studies in the mouse. And this encompassed over 2,000 human samples and 1,600 mouse samples. This included over 100 cell types from the human and about 100 cell types from the mouse as well. So we downloaded all the data from this database called Gene Expression Omnibus, and then fed all that data through the common RiboFlow bioinformatics pipeline to uniformly process all the data. And finally, we looked at the subset of data with matched RiboSeq and RNA-seq pairs. And we QC filtered the samples for high-quality data. And so most of those samples remained. We extracted the signals from this data from mRNA, and then computed the translation efficiency for every mRNA that was measured across many cell types. So these thousands of different samples could then be summarized into two matrices. The first matrix for the mouse was about 11,000 genes or so, encompassing 70 cell types. And the second for the human was about 10,000 genes or so with about 80 cell types. So we first did a preliminary analysis to look at the correlation of translation rates across cell types. So this heat map shows on each column each different cell type and each row a different cell type. And each value here indicates the pairwise Spearman correlation in the rate of translation across many mRNAs in the transcriptome. So red means very high correlation and blue means very low correlation. We can see for the vast majority of these cell types, there's a strong positive correlation close to 0.8, between 0.8 and 1, between many different cell types. But there are a few cell types that are shown here in blue that seem to be outliers at first glance. But we noticed that actually these cell types that clustered poorly with the others are largely those with poorer data quality. So you can see here in this column that many of these cell types have many missing values, so many of the genes weren't properly measured, and the mean coverage of from the signal is too low to actually accurately quantify many of the genes. So we think this is more of a technical artifact rather than biological reality, but by and large, we can see the vast majority of cell types agreeing in the translation rates across cell types. So with this information in hand, we next trained a deep neural network model, we called that RiboNN, to predict translation efficiency from the RNA sequence. So the input to this model is again the RNA sequence from the 5' end to the 3' end. We encode each of those nucleotides again into those 4 different channels, A, C, G, and U, at each position. And we have a last channel that alludes to the reading frame of the open reading frame. So each codon here is highlighted once. So that's all the model can see. And again, we feed it through these convolutions and train a deep convolutional neural network. The output to this model is a little bit different than before. Instead of predicting a single cell type, it predicts all the cell types simultaneously. So all 78 cell types, we call that a multitask model because it predicts many of these tasks simultaneously. The code also for this is available online as well as the pre-trained model, so that's freely available in GitHub and Zenodo. So if you're interested in this work, you're welcome to download the model online. So I want to give you a glimpse into how RiboNN performed. So here in human, on the y-axis, we looked at the mean translation efficiency values across all the cell types for each of the genes in the genome. And on the x-axis is the prediction from RiboNN. We can see here that the Spearman correlation was almost 0.8, meaning that we can very strongly closely predict the rate of translation from the sequence alone. Likewise, in the mouse here, we see that the correlation is around 0.78 or so as well. So in both species, human and mouse independent training models can perform quite well. Next, we looked at different individual cell types. So this is all in human, and we have HMECs down to HeLa cells and kidney and muscle tissue. And on the y-axis is the R squared of the model on the held out data. And we can see there's wide variation in performance. Some of the cell types perform very well, others perform very poorly. In the supplementary figures, we actually showed that those that are performing poorly are the same ones that actually have the lowest quality data, that is the lowest read depth and the highest number of missing genes. So as the sample quality improves, we can actually improve our predictive model as well. Now I've shown you 4 columns here in different colors. The one in green is the base RiboNN model that I mentioned that just predicts all the cell types at the same time. If you just take a single task model, so instead of training and predicting all the cell types at the same time, you predict on that individual cell type, that actually does a little bit better than the multitask model. But if you take the multitask model and further fine-tune it on that individual cell type, then the model can basically learn that same principle and do slightly better than the single task model. Now, what's shown in reddish pink here is what's called a light GBM. It's a classic machine learning model where we feed many different features in and train the classic machine learning model to predict the translation rates. And actually what we found was a little bit different than my prior work. In my prior work, we could usually see that the deep learning model always outperforms the classic machine learning model. In this particular case, it was actually very difficult to outperform the classic ML model. That also performs pretty well. Nevertheless, the neural network still has some value in that it's a convenient all-in-one package to use, and you don't need to compute all of these thousands of features such as a light GBM model requires. So next what we wanted to do was to interpret this model, like ask what it has actually learned. So if anyone in the audience is a wet lab biologist, I used to also be a wet lab biologist. And the philosophical approach when you deal with a cell is that it's a very complex machine. So what can you do with a cell? You can perturb it. For example, you can add a drug to the cell, or you can create a genetic knockout or some sort of perturbation to the cell and then measure the outcome of that. And that's the same thing we can do philosophically with a neural network. So in the same way a cell is a black box, so is a neural network, but you can perturb it and measure what happens at the other end and then learn something about what the neural network has itself learned. So we did that by using several different approaches. One is called attribution scores, and this approach allows you to investigate from the input sequence which regions of that sequence is most important and influential in predicting the outcome. And if you do that across thousands of genes, you can then create a metagene plot to look at which regions of the mRNA have been the most useful to predict translation. You can also use that technique to reconstruct which motifs are most relevant to predict translation. A second thing you can do is to perform what's called in silico insertional analysis. So here you can take a motif that you've discovered and insert it in different positions across the sequence and just ask the neural network what it predicts in different positions after you insert it in different positions. And again, if you do this thousands of times, you can learn something about the positional dependency between the motif and the prediction. So here are the results of the first analysis, where on the y-axis we show the absolute value of those attribution scores. So a higher value means it's more influential or important in the prediction. And we aggregate across thousands of genes to create this plot, segregating genes into 4 different bins, those that have high translation, medium, low, or very low translation rates. And across all these different categories, you can see that consistently the 5' UTR of the mRNA has the highest amount of information in predicting the rate of translation, followed by the open reading frame and followed by a little bit of signal at the beginning of the 3' UTR to predict translation. We observed something pretty similar in the mouse as well, with a similar pattern of this hierarchy of 5' UTR followed by OR followed by 3' UTR. Although the positional impact was slightly different between the mouse and human, the general trend was quite similar. So next we wanted to ask, what are the motifs that are most enriched in regions of high influence or importance? And so I'm ranking the top 5 motifs in the 5' UTR, open reading frame, and 3' UTR in both the human and the mouse on the right. And we see a few patterns emerge. So one is that there's not an obvious major RNA binding protein that sort of stands out. Instead, we actually found that there are these short degenerate elements, even as short as 1 or 2 nucleotides in length, that seem to be the influential ones. So just an example, this GG dinucleotide in the 3' UTR is predicted to downregulate translation. Or a CC dinucleotide in the 5' UTR seems to be common in both the human and the mouse as also downregulating translation. Now, one very well-known motif that was discovered is what's called the upstream and downstream AUG. So we can see in the 5' UTR that this AUG sequence is predicted to downregulate translation of the main open reading frame. But a novel prediction of this model was that even downstream open reading frames are predicted to repress translation. So those that are in the 3' UTR might be repressing translation of the main open reading frame. But this upstream AUG has been very well characterized for about 15, 20 years, that people have known that the presence of these in the 5' UTR, it actually is repressive of translation. So it was really cool to see that the neural network automatically learned this principle without any information or knowledge of this type of motif. Now, knowing that these short degenerate motifs are the most important, we just did a global comparison of all dinucleotides. So UU, AA, UA, etc., all 16 possible combinations. And that's what's shown in this plot on the left. So we insert each of these dinucleotides at different positions across thousands of genes and then measure what is the predicted change in the translation as predicted by our model RiboNN. So here we can see that if we insert a UU dinucleotide, it's predicted to activate translation, both in the human on the left and the mouse on the right. In contrast, if you insert a GG or CC dinucleotide, it's predicted to repress translation in both species as well. And this motif all of these motifs seems to have a spatial dependence. So inserting at the beginning of the 5' UTR, middle, or end can have a different predicted effect depending on exactly where you insert it. So it's possible and seems apparent that this model has learned positional properties of how these dinucleotides and short motifs impact translation. We performed the same analysis now from the perspective instead of codons. So these three nucleotides in the open reading frame, we inserted at different positions along the open reading frame from the beginning of the open reading frame to the end. And we saw again these rich spatial properties. So for example, AGG codon was predicted to repress translation, especially when you insert it very close to the beginning of the start codon of the open reading frame. And in contrast, GCU is a codon having the opposite predicted effect. It was predicted to have an upregulation in translation, especially if you insert it close to the beginning of the open reading frame. And so what we can do now is that we have these rich spatial information, but we can just average that to get the feel, what does the model predict on average the impact of that codon is in both species? And when you do that, you can compute this mean codon influence in the human for the 61 nonstop codons, and also in the mouse on the y-axis for the same 61 nonstop codons. And it was really interesting to see the striking, very strong positive correlation between the species. So these were two independent models trained on species diverged for hundreds of millions of years, yet both models find that the same codons are either upregulating translation or downregulating translation, depending even independent of the species, given that they're both mammals. We've also wanted to also look at whether this principle is related to anything known biophysically about the properties of the ribosome and translation. So we compared this mean codon influence on the x-axis in the human to some previous measurements of like the rate of translation or occupancy in the A site of the ribosome. So this is the site where the tRNA integrates into the ribosome to build the amino acid chain. And how fast the ribosome moves through that is indicative of something about the biophysics of ribosome movement through that codon in particular. So we observed a negative correlation here, meaning that when our model predicted the codon was very much strongly correlated or associated with high translation, it also had the lowest residence time as measured by ribosome occupancy in this earlier study. And that means that the ribosome is very quickly moving through that codon because it's not struggling to decode the amino acid at that position. And a second thing that we looked at is the relationship between our mean codon influence and tRNA abundance. So imagine a cell with many different abundances of one tRNA versus another. You can imagine when the tRNA is very abundant in the cell, it's very easy to stochastically integrate into the ribosome itself. So that's kind of why we see this positive correlation that when the model predicts that there's a strong association with high translation, those are also the cases where the tRNA abundance is high for that particular codon, making it easier to decode that tRNA. I'll show one last result and then wrap up. So because we're in the field of mRNA vaccines and therapeutics, we often use base-modified RNA called, that base modification is called M1 pseudouridine, or M1 pseudo-U. So all the U's are swapped with the M1 pseudo-U, and this experiment from Yale University, they measured thousands of 5' UTRs with this base modification and performed an in vitro translation reaction so they could actually measure the binding of each mRNA to the ribosomes in vitro. And so from this, they computed what's called a ribosome recruitment score, which looks at the binding of ribosomes to each mRNA. So on the y-axis is the measurement of this assay, and on the x-axis is the prediction from our model. We were able to actually see that we could achieve a Spearman correlation of about 0.6, meaning that even with base-modified RNA, our model can still predict something useful about the rate of translation of these 5' UTRs. So now to conclude, have shown you that we have generated a large compendium of human and mouse TE or translation data sets, hopefully serving as a gold standard data for the field. Based on that database, we've developed a state-of-the-art sequence-based model called RiboNN with very strong performance in predicting TEs. And relative to existing models which only consider or largely consider the 5' UTR, RiboNN integrates the entire mRNA sequence. And that allows it to perform a little bit better in predicting the rate of translation. It has learned some interesting position-specific properties about the influence of these short degenerate motifs and codons. And we anticipate the reason we've built these types of models is to better guide the design of translation-optimized RNA for mRNA vaccines. So that's it. Thank you for your attention, and I'm happy to take any questions.
Thank you. Thank you, Dr. Vikram. Really, it is a powerful presentation. I should have asked the first question before the presentation because earlier on, a friend was telling me, but why would someone want to know the translation efficiency of
Yep,
mRNA? And I had an idea why, but unfortunately, she hasn't made it here. She's in transit. But I think let's begin with that question, like why?
sure. Sure, yeah,
Yes.
that's a great question. So imagine there are certain cancers where a particular oncogene is expressed too highly into protein. And in those cancers, there are certain mutations in the sequence of the mRNA that can lead to very high translation of that mRNA. So if we're able to understand the rate of translation, we could actually give insight into the genetic basis of different disorders, including cancer. So that's one type of use of these types of models. You can actually predict for a novel mutation that's never been seen before in the human population. This type of model could actually make a quantitative prediction how much it will increase the rate of translation for that particular mRNA in that particular individual. And that could give insight that this is the underlying mechanistic basis of this mutation that leads to a cancer, for instance. That's just one example, but there are numerous other types of genetic diseases besides cancer that might depend on too high translation. So that's one reason, but the other that I've presented today is to develop therapeutics. We have to design the sequences ourselves. And so if we understand the relationship between the sequence and the rate of translation, then we can design a sequence that will lead to the appropriate amount of translation that we're hoping to achieve from our therapeutic of interest. So those are the two core reasons on the top of my mind so far.
That's great. And yeah, my reasoning was about therapeutic purposes.
Yep,
But yeah, I think that adds up more. Now, doctor, for people who are mainly computer science, of course, data scientists, mathematicians here, maybe you will give a background about codons and maybe as part
sure.
of the enzyme itself. But somewhere I think the paper said, and we also I think said it, like that the codon has positive and negative influence on the translation efficiency. And in my
Yep.
mind, I'm thinking like as a beginner, what should that mean? Like in terms of what is happening at the
Mhm.
ribosome level. Like what
Yes.
does that mean? Yes.
Sure, yeah, let's begin with the first question, which is to define a codon. So here is the diagram of mRNA from left to right. So the codons begin in the coding region, and there are three nucleotides in length. So units of three nucleotides is like what the ribosome recognizes. So when the ribosome scans across this UTR, it begins at the start codon. That's the very beginning of the coding region. And the start codon is AUG. That's the three nucleotide start codon. So that signal tells the ribosome, begin translation here at this position. And every three nucleotides, that each codon is recognized with a tRNA that binds to that codon and then incorporates an amino acid, and it links those amino acids to build the protein. So that's the function of a codon. It's a signal that recruits tRNA to the ribosome to start building the polypeptide or amino acid chain which builds the protein. So that's hopefully defining what a codon is. It's the three nucleotide code in the coding region which specifies what the protein should be translated into at the amino acid level. Now, I mentioned that codons, from when they were discovered in the maybe 1950s or 1960s, that's when they were discovered, and it was discovered that that code encodes the protein. But it took several, maybe even a decade or two, to even realize that codons can have multiple functions. The first function, or the core function, is to be the decoding signal. You can think of it as a dictionary. So you look up this three nucleotide word and it translates into a particular amino acid. So that's a one-to-one dictionary that the nature has created. But the second function of a codon is not just this dictionary to encode the amino acid, but also it governs the rate of movement of the ribosome through the mRNA. So let's imagine a ribosome going very quickly through the mRNA to translate the protein. That's actually been found to lead to RNA being more stable, the RNA being more stable and more efficient protein synthesis. But the opposite is also true. If there's a lot of codons that lead to slow translation, there's a linkage there with fast degradation of the mRNA. So the rate of translation and the rate of RNA decay are actually correlated to one another because of how fast the ribosome is moving through each codon. So those are mechanisms that nature has created that has three meanings. It can regulate the rate of translation, the rate of RNA stability, and also this dictionary that defines what the protein should be made from this mRNA molecule.
Okay. Wow, that's perfect. Members, if you want to ask any question, you can unmute yourself or put the question in the chat. In the meantime, I'm still with Dr. Vikram and still on the codons. So if codons are like a signal and like there could be multiple signals, and clearly that means when these signals are instantiated, they can perform certain influence on sort of like um, on the ribosome or on the enzyme itself. And my question is, would would the two codons, for example, if you have two codons,
Mhm.
and and would they give a different sort of like influence on the result, or
Yep, exactly. So certain codons are associated with tRNAs that there are many tRNAs in the cell that can bind that codon. And because of that, the ribosome can quickly move through decoding that tRNA into the amino acid. But a different codon, even one that encodes the same amino acid, might actually have a different abundance of the corresponding tRNA. That means as the ribosome moves through it, it can be moving through a different rate. And because of that, the rate of decoding can be different, and the cell has a way to recognize the conformation of the ribosome. Like, let's say the ribosome is stuck at that particular codon, it might be temporarily stalled. And because of the conformation of the ribosome, the cell recognizes the ribosome and says, please degrade this mRNA. So that's how there's a linkage between the conformation of the ribosome and that sequence of the codon that leads to one codon being upregulating of translation, and another codon being downregulating of translation.
okay, that's good. I have another question about the ORF and codons, but There was a question in the chat.
Yeah, I think Herbert, you have,
Let me, should I read it for you?
sure.
Okay. Herbert is saying the in silico analysis of motif insertions in the 5 prime UTR was very compelling. Have you performed a similar analysis for a motif that creates a strong secondary structure like a hairpin? That's a question mark. If so, what did the model learn about the impact of secondary structure on translation effect, I think, on
Yep,
translation
that's
efficiency? Yes.
absolutely. Yeah, that's a great question. So I would say that a lot of the research that exists about the relationship between secondary structure and translation, much of that work has actually been shown in bacteria and yeast, so single-celled organisms. I think there's pretty clear and strong evidence that the folding of the RNA sequence in a secondary structure modulates translation. But I would actually argue the opposite, or maybe it's less prominent of a signal in mammalian cells. So in mammalian cells, the ribosome is very aggressively unwinding the RNA sequence. And so that secondary structure that the ribosome has created, it's competing against the ribosome machinery, which it rapidly is unwinding the RNA secondary structure. And so the reason it could impact more in bacteria, for instance, is because the mechanism of ribosome binding in bacteria is very different than that of mammalian cells. So in bacteria, you have the ribosome not actually unwinding the secondary structure. It binds a very specific signal in the 5' UTR of the sequence. And so that's one reason that the mechanism is a little bit different in bacteria versus mammalian cells. But to answer this question more directly, yes, we did look about like one way that we investigated this is a little bit different approach. We looked at all the variation in translation that could not be explained by the primary sequence. So this model is only capturing primary sequence. It doesn't have any knowledge of RNA secondary structure. So what that means is that anything that is unexplained by the primary sequence, we have the opportunity to ask, could secondary structure additionally explain the translation rates of the variation that was unexplained by the primary sequence? So that's the approach that we took. And I can say that when we tried that, we got a diminishingly small difference in the prediction. So we tried like 50 different features to compute secondary structure based on the minimum free energy in the 5' UTR or 3' UTR, different sliding windows. And no matter what we did, we could barely see any difference in the actual predicted rate of translation. Now, it doesn't mean perfectly that secondary structure is irrelevant, because there's almost certainly a strong relationship between base composition and secondary structure. So, for instance, an AU-rich sequence usually has weaker secondary structure than a GC-rich sequence. And so the GC and AU-rich properties that we're seeing might in part be explained by secondary structure, but so far we have not explicitly found that secondary structure is necessary to improve the performance of the model. But it is possible that it has a small and useful impact on translation. Thanks for the question.
Wow, that's fantastic. Um, Doctor, there was somewhere you talked about the ORFs, the open reading frames,
Mhm.
and uh, I think somewhere we saw that inserting codons across ORFs, there was some sort of an effect. I think
Yes.
at the start.
Yeah, that was
Um,
this figure right here.
yes, in insertion analysis. And why is this effect like happening at the start, like it's largest um,
Yep.
at
Great
the
question.
start?
Yeah, we
Yes.
see an effect size that's about double at the start. And there's actually a very specific reason for that. So in regulating translation, there's two phases. There's the initiation phase called translation initiation. That's when the ribosome recognizes the start codon and begins translation. And then the second phase is after translation has begun. That's called the elongation phase. So it continues elongating through the entire sequence. So generally speaking, the first part, initiation, is the core rate limiting step or bottleneck that prevents downstream translation. And initiation happens
Okay.
right in the first 10 to 15 codons of the open reading frame. So our belief is that the model has actually picked up that these codons have a much more important effect in translation initiation and getting the ribosome moving from the beginning than it does later in the message when the ribosome is already moving. So that's the key
Okay.
mechanistic reason we believe that there's this greater impact in the beginning.
No. Okay. I think from me, the last one would be, uh, Doctor, when you look at most research in regards to um, trying to deal with proteins and uh, mRNAs, there is a tendency to use um, models close to LLMs,
Mhm.
and and basically, well, it would make sense because we are looking at sort of like the chains. We can look at them as language patterns, maybe.
Yes,
And your choice was this close to a CNN, the
true.
RiboNN, if I'm right, and no fancy things like attention and all that.
Mhm.
Um, and we know, like, whenever we think about RNNs, what comes to my mind is images, things close to the image analysis.
Yep.
So why the choice of the neural network?
Great question. So basically, CNNs are really great at capturing local properties. So when you have a string of nucleotides all in a particular like configuration, like for example, UCGU, that's a four
Yes,
nucleotide pattern that if you used attention, it would have to look at all by all, like a 4 by 4 matrix of
true.
attention,
Yes.
right? So, but a
Mm.
CNN can very efficiently capture that pattern in a single convolutional kernel.
Oh yeah.
So it's much more parameter efficient, and the training speed is much faster too. That being said, you can do hybrid things. You can use CNN layers followed by the transformer layer. That's one possibility that we've trained in the past. Or
Okay.
you could use the all attention layer, just the transformers, with several stack of transformer layers. So that is possible, but it's extremely parameter hungry, especially when you get to thousands of nucleotides. Something that's like 5,000 nucleotides, the all by all attention that quickly explodes in the n by n matrix that you have to construct as the
Yeah.
length of the sequence gets longer and longer. So
True.
the CNN is something that's very parameter efficient, is very fast to train, and it often performs very well in these tasks already. So I kind of think of it as a baseline that like it's a great starting point. It doesn't mean that we need to stop here. You can always try CNN hybrid, like transformer hybrid networks as well. But usually in experimenting with these alternative architectures, we haven't really seen that the other ones do much better or even better than the simple CNN architecture. So that's why we have preferred like let's start simple with something that's fast and efficient to train.
Perfect. That clears my curiosity. Innocent, your hand is up. You can unmute yourself if it wasn't a mistake.
Okay, thank
Um,
you.
okay.
Now I want to ask something
Yes,
about
sure.
the kinetics because initially, when the presenter, when he was starting, he demonstrated something like he showed a messenger RNA with many ribosomes attached, and then another messenger RNA with one ribosome attached. And what I picked in the beginning was like a rate or high rate of
Like this one.
translation leads to high, yeah, like high rate of translation leads to higher transcription efficiency.
Yes.
That's what I picked at the beginning.
Higher
So in that case,
translation efficiency, yes.
I'm looking at other factors because before the amino acids are brought there to the ribosomes to start polymerizing those amino acids into protein, there are other factors like amino acid activation, which require ATP, and then after a transfer, RNA will bring it to the ribosome for the assembly. Now, but in the presentation, I'm seeing that maybe you are focusing on the number of ribosomes attached to the messenger RNA. Like, how did you manage to control other factors, like maybe because not because I'm thinking about like maybe those ribosomes may be there when there are very many, maybe when they are still waiting for the amino acid to be brought by the transfer RNA, which may sometimes delay to bring because there are other processes which require energy to be loaded on the transfer and then to be brought there. Like, can those some of those ribosomes can they be there when they're just doing nothing? They are just waiting and then they slow down the process, and maybe you may find that the other one, which is on a single on one messenger RNA, one ribosome and a messenger RNA, for it is active, or yes, like the rate at which the transfer RNA are bringing amino acid is faster than when these ones are just reactant. So how did you manage to optimize those conditions such that maybe you are focusing on the number of ribosomes attached on the messenger RNA?
Yeah, that's a
So
very
that
important
is it.
question. So first of all, a single mRNA can have multiple ribosomes loaded on it at the exact same moment in time. So that's called like a polysome because they're like 4 or 5 or 6 ribosomes all translating simultaneously. So that's why this diagram on the bottom shows that. Now, this field is a little bit controversial. There are historically some people have said that the rate of elongation is almost constant from one message to another. So it doesn't really matter from their perspective the composition of the sequence of messenger RNA 1 versus 2. This research actually shows that we predict from the model that there are slight differences, a very diminishingly small difference between one codon and another, but collectively when you have thousands of those codons linked together, that can govern the overall rate of translation. Now you're right that when ribosomes are moving to the start codon, there is a temporary stall there, and that's why I alluded that there is a bottleneck in translation initiation. That if there's a ribosome frozen there, upstream ribosomes cannot load and begin translating because it's kind of stalling and blocking the rate of ribosome like the initiation process to move into elongation. So that's why people usually allude to that as the rate limiting step of translation itself. And there are signals in the 5' UTR and early part of the codons that impact whether that ribosome can start beginning the elongation process. So in our measurement, we're actually normalizing to the length of the sequence. So you might be worried that a long sequence might be having lower number of ribosomes, but we're actually looking at the density of ribosomes no matter what the length is, because we normalize the riboseq data to the RNA-seq data. And when you do that normalization, both of those are of the same length, which means the length term cancels out. And then you're just looking at the density of the ribosomes on the sequence. So hopefully that kind of gives, I don't know if I perfectly answered your question or fully understood it, but hopefully that gives you a sense of what we're measuring and why the initiation is the core bottleneck.
So, Innocent, I hope the response was satisfying. Okay, if there are no other questions, I want to come to the top of the session by thanking Dr. Vikram for saving up his time on his tight schedule and really talk to us. I want to thank everyone who has attended this call.